honor for the graduate program in biophysics from the Carlos Chagas Filho Institute of Biophysics from the Federal University of Rio de Janeiro to have you as a speaker in our discipline of our program. You are very welcome to our institute, and we want you to feel at home. And of course, we are going to learn so much about your talk, about your work here. And for our students, it's a pleasure to listen to you today. Thank you very much. Please feel at home and be very welcome to our institute. Thank you very much.
Thank you.
Okay. Can I start? Are you hearing me?
Yes, Vanderlei.
Okay. Well, it's a great pleasure for me to introduce Hugo Luján, that will talk today about using protozoa surface proteins for effective oral vaccination. Hugo, it's an old and good friend. He made his undergraduation and the master degree at the National University of Cordoba in Argentina. Then he moved to the United States on the stage for 5 years at the NIH doing a very important work on Giardia intestinalis. I forgot to point out that his first work was in Trypanosoma cruzi. That was the topic of his master's degree. But his studies in Giardia lamblia or Giardia intestinalis are very well recognized throughout the world. I would say that Hugo is amongst the best scientists work with parasitic protozoa, always innovating in approach and innovation in the results that he obtained. Since after the NIH postdoc, he came back to Argentina and established again in his original university as an assistant and associate professor of the Universidad Nacional de Cordoba. But then he was invited and accepted the position of full professor of biochemistry and cell biology at the University, Catolica University of Cordoba, Argentina, where he also established a very nice group. I had the opportunity to visit his laboratory a few years ago, and he is a director of the Centro de Investigación y Desarrollo en Inmunología e Enfermedades Infecciosas del CONICET en la Universidad Católica de Córdoba, Argentina. His research has given important contributions to the knowledge on differentiation of Giardia, the synthesis of the cis-wall proteins, and the assembly of the cis-wall, synthesis and regulation of the variant-specific surface proteins, and more recently, using a bioengineering approach, he's using these VSPs as a platform to produce virus-like particles for oral vaccines. I knew last week during a symposium of the National Academy of Medicine that he's also involved in cooperation with Brazilian friends in the development of a vaccine for SARS-CoV-2. And this means that he is using this platform to do an innovative approach in vaccines in different pathogenic microorganisms, so that we have a collaboration between our labs and the Hugo Luján labs that is going on very well, and I hope we will increase in the next few years. It's a great pleasure for me, Hugo, to give you this opportunity that to talk not only for the Biophysics Institute, but through the YouTube. People throughout the world are participating of this symposium. Please.
Thank you, Vanderlei. It's a great pleasure for me, in particular, the celebration of the 75th anniversary of the Biophysics Institute at the Federal University of Rio de Janeiro. It's really good. I just can say that you spoiled all my talk, but I will try to make a story here to show your students and students around the world how we can go from very specific basic research, fundamental research, to applied research. When someone is able to discover the mechanism of the biology of the cell that somebody is studying. So I will start with the story. I still call in this parasite Giardia lamblia. Giardia lamblia, as you may know, inhabit the upper small intestine of many vertebrate cells. You can see here this old picture that you can find in every book of parasitology, in which Giardia have this happy face with the two nuclei looking like eyes, the medial body, the flagella. So it's a nice cells to work with, and you know that very well. It's transmitted by contaminated water or food, and this Giardia trophozoa may differentiate into cysts, and those cysts can remain viable for years outside the host. So most of my career has been devoted to understand the mechanism of Giardia trophozoa differentiation into cysts, but also another mechanism called antigenic variation, in which this parasite continually change the surface antigen in order to avoid the host immune response, as we will see later. But the most important point is that Giardia can, I'm showing now a picture taken by Vanderlei and Victor, showing Giardia in culture, and you can see that the parasite has a particular organelle called ventral disc. This ventral or sucking disc can attach very strongly to the surface of the intestinal mucosa and remains there, causing the disease called giardiasis. One big question is, how can Giardia survive in such hostile environment? The upper small intestine is the site of the body where digestion takes place. A lot of secretion coming from the liver, the pancreas, release enzymes and compounds that make us able to digest macromolecules for further absorption. And Giardia appears to be the only microorganism able to colonize the lumen of the upper small intestine. And how can Giardia survive in that environment is really, it was a tough question for years. From this beautiful picture of Paulo Pimenta, a Brazilian researcher at the NIH in 1989, he published this very nice picture showing how antibodies labeled with gold particles are directed to surface antigen of Giardia called VSPs for variant-specific surface protein, stain the entire surface of the trophozoa, including the ventral disc and the flagella. These surface antigens form a dense coat on the surface of the parasite and make most of the other molecules present in the surface unable to be seen by the host immune response. So this coat formed by VSP looked like a protective code for the parasite in order to survive on the intestine. And that was the hypothesis for years, but remains that way also for years. These proteins also undergoes the process of antigenic variation. These Giardia VSPs are molecules that cover, like I said, the entire surface of the parasite. This is an immunofluorescent imaging. You can see that a monoclonal antibody directed to only one of these surface antigen only labeled some cells, while others remain unlabeled, meaning that this parasite is able to switch the surface antigens among members of a family of homologous genes present on the parasite genome. For years, antigenic variation in parasitic protozoa has been regarded as a mechanism for evasion of the host immune response. However, no one was able to disrupt the mechanism to see if that theory was true. And you will see that during many years of studies of Giardia, we were able to perform some studies that presented some light into the system. One of the characteristics of this protein is when, so far no one was able to crystallize or to obtain the structure of these molecules, but some prediction software show that these are very disordered molecule with a very few beta sheets and alpha helixes, but most of the protein is really very tough. And if you make this prediction, this modeling with different VSPs from this family, you will see that most of them look like this. Very difficult to understand how a protein with this characteristic can be able to protect the parasite. So Giardia VSP not only protect the parasite from digestion in the upper small intestine, but also allows Giardia to evade the host immune response. These VSP have the following characteristics. These are type 1 membrane proteins that contain a C-terminal domain, a very conserved C-terminal domain, including a transmembrane region and a very short cytoplasmic tail that contain only 5 amino acids, CRGKA. And this tail, cytoplasmic tail, is present in all known VSPs, but also have a very large extracellular domain, extracellular region, which is rich in cysteines, approximately between 15 and 17% of cysteine in this ectodomain of the protein. So this is the area of the protein that can be seen by the host because they form the interface between the host and the parasite. The molecular weight of this protein varies between 40 and 150 kilodaltons, and this region, rich in cysteine, contain more. The presence of cysteine in this domain is mostly in this motif, a cysteine XX cysteine motif, in which X can be any other amino acids. I was the first one to purify one of the VSPs when I was at the NIH in the 90s, and we found that these motifs are able to bind iron and zinc. So the binding of metals to the VSP may confer the protein this specific characteristic to be resistant to proteolytic digestion in the upper small intestine. Another thing is that the VSP are highly immunogenic. It's very common to find in the serum of a patient or of any person specific in endemic areas, that have antibodies against Giardia VSPs. So that makes this protein really, really immunogenic, and this is one of the important points. Not only that the VSP protect the parasite from digestion in the upper small intestine, but also trigger a very strong immune response to them. Like I said before, the antigenic variation from this 150 VSP coding gene present in the parasite genome, only one VSP is expressed at any point in time, and the switching from one to another is supposed to be spontaneous after a given number of generations. In 2008, and this is something that I want to show, in particular the students, is how we went from this basic fundamental research on the biology of Giardia to what we are working now. And it's a very nice story. We published in Nature in 2008 that antigenic variation in Giardia is regulated by RNA interference. That means most of the VSP genes are transcribed simultaneously on parasites, but molecules of the RNA pathway present in the cytoplasm of the cell recognize these homologous genes, silencing all but one of them, and this one that is not silent accumulates and is translated into the protein that then is going to be expressed on the surface of the parasite. But I just want to point out that a paper in Nature is very hard to publish from Latin America. And importantly, I want to show that if I could do it, everybody else can do it. In particular, I'm the only one in the list of authors of this paper that was a PhD at the time. All of the other students were PhD students at the time, and they have a paper published in a top journal when they were around 25 years old, something that all researchers wanted to have for sure. In that paper, we showed that Giardia, from expressing only one VSP on the surface, was able to express more than one at the same time, simultaneously. This is a monoclonal antibody against a VSP called 9B10, and this is a monoclonal antibody to a VSP called 1267. As you can see, from expressing just one, as I showed in previous slides, in here, after disrupting antigenic variation by knocking down the expression of key enzymes of the RNA pathway, like RNA-dependent RNA polymerase and dicer, we were able to obtain parasite obtaining many VSP at the same time on the surface. That was mostly proved by the use of a particular monoclonal antibody that we developed that was able to recognize these 5 amino acids that are present in the cytoplasmic tail of all VSP. So the most conserved domain of all the VSP is this 5 amino acid cytoplasmic tail. We were able to develop a monoclonal antibody to it, and by Western blot we saw that when silenced dicer or RDRP, we were able to see that many VSP are being produced and expressed on those cells in comparison to the one that there is not, in which antigenic variation was not disrupted. So for that point, we were able to show that disruption of antigenic variation was able to produce a modified parasite expressing the entire repertoire of VSP on their surface. And then we decided, okay, we have these cells that have all the VSPs, so they are not variant anymore. Can we immunize animals to see if they are protected against the infection by Giardia Cs or trophozoans. And a couple of years later, we were able to publish this paper in which it's entitled "Disruption of Antigenic Variation is Crucial for Effective Parasite Vaccine." Why? Because we were able to infect gerbils. Giardia lamblia is not able to infect mice. Mice are infected by a related species called Giardia viris. So we have to use another animal model. A good animal model is Meriones ungiculatus, the gerbils. And in that case, we're also able to show that if we vaccinate the animals, we get a good immune response that protected the animals for subsequent infection with Giardia. But one important point here is that we use this full repertoire of Giardia VSP for vaccination, but we did it orally, thinking if a cyst of Giardia, which is no other thing that a trophozoid enclosed into a cyst wall, if these VSPs on the cyst were able to recede for even years on a school table, we would be able to vaccinate orally. And we did it. We vaccinated orally, and we found that by that means the gerbil was fully protected to subsequent infection with any Giardia expressing many different VSP. In particular, VSP that we don't know because they came from cysts taken for a patient from the clinic. And remarkably, the immunization was performed in the absence of adjuvants. What was something really good, because many people that are against vaccines are not against vaccine per se, are against these adjuvants that some people claim that cause some crazy things. Another important thing was that the gerbil didn't present any signs of the disease. So it's clear by the experiment that we published in 2010 that the Giardia parasite is what caused giardiasis. The VSPs are not toxic to cells or to animals, and we demonstrated in this work. So the VSP can easily be used to vaccinate animals. Later, we used this vaccine formulation consisting in the full repertoire of VSP to vaccinate domestic animals. We vaccinate dogs and cats, and we showed that this novel oral vaccine prevents Giardia infection, but also alleviates the signs of giardiasis in the infected animals. And when we tested the vaccination in dogs living in, running freely in a shanty town here in Argentina, we saw that vaccinating the animals, we can reduce the transmission to humans. When we started this work, almost all the children of that village were infected, where 100% of the children were infected for giardiasis. After a year or so, that infection rate decreases down to 30%. So we can not only show that Giardia is a zoonotic infection, but also if we vaccinate animals, we can prevent infection of humans in that case. So oral vaccination using VSP generated a strong immune response that protects gerbils, dogs, and cats against Giardia parasites. And that vaccine was licensed by CONICET to an international company that pretend that it's going to be available soon. So after seeing that the VSPs of this VSP, which are cysteine, CXXC-rich molecules, if we wonder if they are unique to Giardia parasite, or other parasites also have these molecules on their surface. And here you won't see the sequence, but you will see in green this motif, the cysteine X cysteine motif. In yellow, you will see free cysteines. And we have a VSP of the VSP-9B10 from Giardia assemblage A, VSP-1267 for Giardia assemblage A2, and here Giardia VSP A7, which belong to the strain called GS, belonging to the assemblage B. So as you can see, most of the VSP have the same presence of a numerous motif containing cysteine, any other amino acid twice, and then another cysteine. But if we look at surface molecules of other parasites like Entamoeba histolytica, we can see this protein kinase. It also contains numerous cysteine X motifs on the surface. But also free-living organisms like Paramecium or Tetrahymena, which are not infection. They live in water and everywhere. So they also have this motif on their surface. And they also always have some other free cysteine on it. So we decided to see if this the presence of the cysteine XX cysteine motif were essential to allow this protein to have these particular properties. And what we did was we saw first that all of these microorganisms live in harsh environmental conditions. Entamoeba in the large intestine, Paramecium and Tetrahymena, they live with change in temperature, osmotic force, and osmotic strength, and changes in pH, etc. So we thought at the time that this protein will be important in protecting this microorganism. So here also it's going to be hard for you to see, but we use Giardia on the top, Entamoeba histolytica, Tetrahymena, Paramecium, and mammalian cells. And we just grew them in culture medium or in PBS, and you will see that they don't change much. When we treat them with trypsin, protease, which is released to the intestine from the pancreas, we can see that most of the parasites are fine, but the mammalian cell start looking really badly. If we increase the amount of this trypsin, we can see that Giardia, Entamoeba, Tetrahymena, and Paramecium remains fine, but most of the mammalian cells are destroyed with this trypsin concentration. And that can be seen better if we include fluorescent diacetate to stain living cell and propidium iodine to stain the nucleus of dead cells. And you can see that Giardia, Entamoeba histolytica, Tetrahymena, Thermophila, or Paramecium tetrahymena, all of them remain viable with high concentration of trypsin, but NSO cells we use to produce monoclonal antibodies, they grow in suspension, they are totally destroyed, and their nucleus is stained by propidium iodine in red, showing that this microorganism having cysteine-rich molecules on their surface may resist a proteolytic digestion better than mammalian cells. Then we decided to produce these VSPs in high quantity and quality in the laboratory. And for that, we took away the transmembrane domain and the cytoplasmic tail of the VSP, and we call them delta VSP. This only consists on the ectodomain of the VSP with a histag for purification, And we produce these VSPs in insect cell lines or in mammalian cell lines to get high amounts of this protein for further experiments. But first, we saw that these delta VSPs that were confronted to very harsh pH, pH2, pH10, or protein trypsin ratios that vary, including when we confronted with intestinal extract of gerbils, we can see that most of the proteins remain stable when other protein, like any other antigen, like the HA from the hemagglutinin protein from the influenza virus, we can see that with this drastic condition, it are completely destroyed. We also use stomach extract to see what's they have any effect on the VSP, and we can see that the VSP remain fine, whereas the HA is destroyed. And that just is to show the cytometry of this Western blot to better see how is the difference between the resistance to harsh environmental condition by the VSP in comparison to any other antigen like the flu, hemagglutinin. We don't not only use VSP-1267 or 9B10 for this kind of studies, but also the VSP-A7, which belong, like I said, to the GS isolate from the assemblage B. But also we tested how the trophozoite behave, expressing on the surface the VSP-1267, A7, or 9B10, and in all the cases remain very, very stable. It's different with a very strong antigen of Giardia. It's called BIP. It's a chaperone endoplasmic reticulum, which can produce a very strong immune response too, but it's intracellular, and we can see that it's destroyed under some of the conditions that we use for the VSP, in which the VSP remains very stable. So after knowing that the VSP conferred this resistance to proteolytic digestion and different pH and different environmental, very tough environmental conditions, we wonder, why the VSP are highly immunogenic. And for that, we recognize initially that signaling through the toll-like receptor could be a good way to signal by these molecules. And so we initially use a mammalian cell line, a reported mammalian cell line with stable, expressed on the surface, different toll-like receptors. In this case, they were human toll-like receptors. And we confirmed these cells to purify VSPs. And we saw that this signal is activated by in toll-like receptor 2 and toll-like receptor 4. Like I said, this is our human. So then we're trying to use mouse to see if that it was a case. And really, if you can see here in red is the toll-like receptor 4 reported cell line, and in blue the toll-like receptor 2. The activation of the receptor is dose dependent. Is higher for toll-like receptor 4, and you can see there is a huge difference with toll-like receptor 2. So we were really clear that the VSP are able to activate toll-like receptor 4, and that may cause this very strong immune response, because these receptors are key in activating the adaptive immune response. And any other protein, like the hemagglutinin of the flu virus, we can see that they don't activate these receptors. The same could be observed when we look at dendritic cells obtained from wild type or toll-like receptor for knockout mice, in which we look at the presence and the activation of co-stimulatory molecules like this CD40 or CD86, and we can see that the VSP-1267 is able to activate these molecules and the wild type, but not in the toll-like receptor for knockout mice. So this protein containing multiple system XX system motifs can be considered a novel pathogen-associated molecular pattern acting via the toll-like receptor 4. So at that point, knowing that we have good protein for oral delivery of molecules, and at the same time, molecules with adjuvant properties, because they activate our receptor 4, we wonder if we can use this VSP to protect an adjuvant heterologous antigen for oral administrations. And we all know that the first successful vaccine were live attenuated or heat inactivated pathogen. That means particle-based formulations. So together with a group in France, which was Dr. David Klassmann, we decided to use virus-like particles. And virus-like particles are really common for vaccination. Nowadays, these VLPs for virus-like particles are used for vaccination against hepatitis B or for human papillomavirus virus. These molecules, these particles can be easily produced in the laboratory and even in industrial way, just by expressing molecules of some viruses in order to assemble like a regular virus without having the nucleic acid of the virus. So these particles are totally ineffective. So we decided to use retro VLPs in which if you use the gag protein of any of these viruses and express them in mammalian cells, this molecule can assemble into the capsid by themselves spontaneously when produced, and then back from the cells to taking with them the envelope which consists in the plasma membrane of those cells. So by typical molecular biology techniques and cell biology techniques in the lab, we can produce these viral-like particles just by using parts of different viruses. But we decided to use the VSPs of Giardia for protection and adjuvantation of these VLPs, because these VSPs, the naked or the plain VLPs, were that even those that are in the market now are given by injection. So we thought if we can include on the surface of the VLP the ectodomain of the VSP, we can easily produce particles that can be administered orally to individuals. Like making a nanogiardia in which we have the protection and immunogenic properties of the VSPs on the surface of them. And we decided to start with a very common system, which is the influenza virus as a model. You know, the influenza virus have two major surface proteins. One of them is HA, the hemagglutinin, and another one is neuraminidase. So hemagglutinin can bind very strongly to sialic acid on human cells, and but the neuraminidase is necessary to release the sialic acid of the infected cell for the release of this virus to the environment. So these two molecules were included on the VSPs just by combination of many different molecular biology techniques. So we use the only the ectodomain of the VSPs fused to the transmembrane domain and the cytoplasmic tail of the VSPG protein. So this protein is known to attach to GAG from the murine leukemia virus very tightly. So when the capsid forms inside the cell by the expression of GAG, can take with them the VSP fused to this cytoplasmic domain of the VSPG. And we use these two antigen of the influenza virus, we said, the HA, the hemagglutinin, which is also a type 1 membrane protein, and neuraminidase, which is a type 2 membrane protein. And importantly, you can fuse to GAG any other protein in order to also add another antigen to the formulation. But in this case, we use the yellow fluorescent protein in order to see the particle. So in that case, we have two VLPs, one that we call naked or plain VSP, and the other which is protected by the VSP, that contain Na and HA on the surface and GAG fused to yellow fluorescent protein in the interior of the particle. And here you can see that, that's a control to show that all the proteins are easily produced, and in mammalian cells we use HEC 297 cells, 293 cells for the production of the VLP in culture, and we tested these molecules for the expression not only of the VSP but also by the HA and Na in the surface, which this protein can be easily pseudotyped on the molecules. For validation of these vaccinal VLPs, we also perform Western blot to see if HA is present, if the Na is present, of the VSP is present only in the one in which will transfect, that is not the plain VSP, but is the pseudotype with the VSP, and GAG also present in these particles. And we tested if HA was correctly folded on the surface by validation by the inhibition of the hemagglutination assay. And we can see here that both the plain VLP and the pseudotype, VSP pseudotype VLP, were able to inhibit hemagglutination better than even the current vaccine, which is the Viraflu. We also were able to measure the size of these particles, and we saw that the plain VLP have 140 nanometers of diameter, and the VSP, the one that contains the VSP, is a little bit larger. We tested if these VLPs were also resistant to the same assay that I showed you for the purified VSP, observing exactly the same results. The VSP are protected, but now we can see that the HA from the VLP that doesn't have the VSP are easily degraded, but the one that already have the VSP on the surface remains stable. So that means that the presence of the VSP on the surface of the VLP can protect heterologous antigen from degradation. We measure also these immunogenic properties of these particles, and we saw again that the activation of CD40 or CD86 on the wild type of the knockout of the toll-like receptor 4 knockout mice, and we can see that in the presence of the VSP, the VLP produce a high activation similar to LPS. The same happened with the other molecule. We then studied in animals that were vaccinated with these particles, the production of TNF, interleukin 10, interleukin 6, and again, those particles having the VSP on the surface were able to strongly activate the expression of the cytokines, the inflammatory cytokines, when given orally to mice. And again, because that didn't happen with in the knockout mouse, we can see that this VLPs pseudotype with VSP signal through the toll-like receptor 4. So also to continue studying the immunogenic properties of purified vaccinal VLPs, we measure the antibody production in serum against two strains of influenza virus, the H1N1 and H5N1, and we saw that the production of serum IgG is higher in those vaccinated with the protected VLP, the same with H5N1. IgG1 happened the same for the two conditions. And N5 with IgG1 or IgG2A, also they have a better production of serum antibodies of all classes. And the production of these immunoglobulins, the heterologous antigen, the hemagglutinin of the virus, increases during the time. These are days for post-immunization, and we can see that even in mice, this high titer of antibodies remains high after 125 days. Now, if we look at the serum titer of IgG against HA of N5N1, we can see that the plain VSP produce a higher titer of antibodies in this case compared to our VSP, and that could be true. But the question that the production of antibodies systemically it had nothing to do with the production of antibodies on the mucosa, which we know most of the pathogenic agents enter our body. So when we measure immunoglobulin A, we saw that in that case, in the feces of these animals, the titer is higher with our formulation than with the plain VLP. And the same happened with the immunoglobulin A secreted into the bronchoalveolar lavage of these animals. So we saw a higher titer of antibodies in the fluids of the vaccinated animals in comparison to the unvaccinated or the one vaccinated with the plain or the naked VLP. The interferon gamma production was also higher with our preparation, and again this inflammatory cytokines increased better with the pseudotype VLPs that the plain VSP. Another important thing was the fact that the VLP pseudotype with the Giardia VSP or any other molecules containing cysteine, XX cysteine motif produce higher titer or neutralizing antibodies against HA in comparison with unvaccinated animals or the plain VSP. So that made very clear that this way of immunization produces a neutralizing antibody, which are better. And another thing that we did is just to show that even if we keep this vaccinal formulation at -70, -4 degrees at room temperature, after 10 cycles of freeze and thaw, so they keep having the same immunogenic properties, meaning that like the Giardia cyst or the Giardia VSP, this molecule are not destroyed by different condition of storage or distribution. So VSP pseudotypes VLP are resistant to the digestion and changes on temperature and pH. So we have antibodies, we have a good immune response, but these responses were protected. We didn't know, so we have to challenge these animals with a mouse-adapted strain of influenza, and we can see here that only the animals vaccinated with the preparation, the oral vaccination with VLP containing HA and pseudotype with the VSP truly protected the animals from the viral challenge. While the other condition, you can see the decrease and the death of the animals at early times. Only the vaccine, the commercial vaccine, allows some of the animals to survive the viral challenge. So VSP pseudotype VSP are affected at distal mucosal sites. So remember, we vaccinated orally, so we vaccinated into the upper small intestine, but we have an immune response for the lungs of these animals that were challenged intranasally with the live virus. So in addition to look for antibodies, we're trying to see if we can have a cytotoxic T-cell response to using our oral vaccines. And for that, we use some tumor cells expressing on their surface the flu antigens. So these are a mesothelioma tumor in which we can express the hemagglutinin molecule from the influenza virus on their surface, and we can inject those cells into mice that were previously vaccinated or not. And we can see that in the naive mice, the tumor that these cells produce are huge after some days after the inoculation. If we use animals already vaccinated with naked VLP, the tumors were also large, but we orally vaccinated with the VLP HA pseudotype with the Giardia VSP, and only one of the mice we can see a very small tumor, while in the other one, the tumor was not able to be induced. And in the presence of the VLP HA plus an adjuvant and immunized by injection, the size of the tumor are mostly like what happened in the unvaccinated animals. This is just to show that the tumor size and the tumor weight were increasing a lot in the presence of in the animals vaccinated with formulation other than the VSP VLP, in which we cannot see any tumor. So we also check the presence of immunoglobulins and interferon in those cells, as indicating that the production of a cellular immune response was also produced, and not only a humoral immune response was produced by oral vaccination with these VSPs. Looking at those so amazing results, we decided to see if we can therapeutically vaccinate animals that already were expressing, or already were having the tumors. So we use in this case the HA-Mesothelioma tumor, but also HA breast cancer tumor, the 41 cells. Remember, in those cases, these cell lines express on the surface the antigen of the influenza virus to which we vaccinated. Unfortunately, for the mesothelioma tumor, we were not able to stop the growing of the tumor vaccinated starting at day 20 after inoculation of the tumor cells. But with the breast cancer tumor, we saw animals immunized with the oral preparation of the VSP. So this VSP pseudotype VLP may be used an anti-cancer vaccine as well. We are not focusing on that at the time, but it's something to look at, just thinking on personalized medicine and taking a tumor, looking at new antigen, and just formulating this vaccine in the lab to treat some patient is something that we always keep in mind. So we came from studying the antigenic variation and adaptation of early branching eukaryotic cell like Giardia to produce this nano Giardia viral particles that can carry heterologous antigen for oral vaccination. And just to summarize that, the VLP VSP are important for that. They are resistant to acidic pH, proteolytic digestion, and changes in temperature. So they don't require refrigeration for storage or transportation. They elicit a strong immune response against any antigen, in this case HA, when given orally, and they generate a protective response to live virus, sorry, stay here. And both neutralizing antibody and cytotoxicity cell are generated. And similar result we obtained with VSP-like domain from several other microorganisms like Paramecium, Tetrahymena, and Tetramen. We were able to publish this last year in Nature Communication as efficient oral vaccination by bioengineering viralized pyrethroid with protozoan and surface protein. Let's highlight the importance of the upper small intestine as a potent target for effective vaccination to get both mucosal immunity and systemic immunity. They may also play a significant role in confronting vaccine refusal and hesitancy due to the aversion to needles and discomfort to metal and chemical-based adjuvant and so on. So we keep working on that. We work in progress where we use antigen of dengue, Zika, respiratory syncytial virus, Venezuelan encephalitis virus, etc. And at that point, the novel coronavirus came in. And if we were having a good platform for the design of oral vaccine, we decided to jump into it. And as you know, the novel coronavirus SARS-CoV-2, the major protein is the spike protein on the surface that have two conformations, mostly the pre-fusion conformation and the post-fusion conformation. And the pre-fusion conformation can bind to the receptor binding domain. This receptor binding domain can bind to the AC2, the angiotensin converting enzyme 2. I'm sure more of you know the virus now. So I took this model from the scientists that was published, in which you can see the movement of this molecule, the spike protein, in which only when the prefusion can bind to the AC2 site. So when stabilized in this position, you can generate neutralizing antibodies to this to this region of the molecule that can make inhibiting and can inhibit the binding to the receptor at the mucosal surface of the body, so preventing the virus entry into our body. So we developed similarly to what we did with influenza, but in this case, expressing the spike protein, stabilized using many different approaches, but also we can fuse to gag, in this case, portion of the N protein of any other ORF encoded by the novel coronavirus genome in order to foster the generation of cytotoxic T cells. So this versatile oral vaccine platform based on VSP pseudotype VLPs can be easily adapted to different antigens for different infection agents and tumors, and have the characteristic to help reduce resistance to vaccination, which can facilitate mass vaccination program around the world, in particular to be used in remote areas of the world where vaccine refrigeration is really impractical. So I just have to finish. I just want to thank the people working in my group, including Renata Tunelli from Sao Paulo, that helped us a lot during the work with the influenza virus, David Clathman in Paris, France, and Dr. Tim Esper-Wasser group in Mainz, Germany. And now with the novel coronavirus, we're working in collaboration with Dr. Jorge Khalil at Sao Paulo. We also appreciate the financial support of CONICET, the Ministry of Science and Technology in Argentina, the University, the Alexander von Humboldt Foundation from Germany, and CompuVac program from the European Union, and several donors that are collaborating now with us to develop faster coronavirus vaccine. Thanks a lot.
Hello, Dr. Luján. Are everything all right? Can you hear me?
Sure.
My name is Miriam. I'm from here, Institute of Biophysics, from University of Rio de Janeiro. And I'm here to help you with some questions for our public. We have questions from YouTube and from here, from the Google Meet. Could you help us?
Sure.
Okay, so let's talk about the first question from Marcia Thiers, professor from here, our lab. What would be the meaning of the simultaneous expression of two VSPs? As the trophozoite divides, one antigen will gradually replace the previous. Does Giardia do surface shedding of VSPs as seen in other parasites such as T. brucei, Trypanosoma brucei?
You know, from the 150 VSP genes encoded in the Giardia genome, only one is expressed on the trophozoite surface. The switching of that to another one may occur every certain generation, that was told for many years, or also can be triggered by certain stimuli. We're working on those stimuli, and we can see that there are two options. One, if the antigen can be endoscopic, or can be released into the culture medium or the environment. We are working on that.
Okay. Next, it's from Professor Vanderlei de Souza. Could you comment on the role played by the iron and zinc bind sites in the VSPs? How these irons interfere with the protozoa?
Well, iron and zinc are essential micronutrients of Giardia, no doubt about it. Now, in relationship to the VSPs, we published a paper in Journal of Biological Chemistry in, I think it was 1996, in which you can see that this system, XX system motif, can coordinate a metal zinc or iron, and in that form produce the structure of the protein, which can be modeled by some computer software. You can see that it's very distorted. But the fact that you have metals working in getting the right conformation of the protein, that means that these proteins are more easily, more difficultly sensitive to degradation. I didn't show the results during my presentation, but you can find in the Nature communication paper that when we chelate or block these disulfide bonds or the coordination of metal, we can disrupt these properties of the VSP. Not only the resistance to proteolytic digestion, different pHs, etc., but also the immunological properties of the VSP are lost when we treat it with strong chelators and with drugs that disturb the redox state of the protein.
Okay. From YouTube, Dr. Vitor Midlash, working in Fiocruz, and the question is, there was a Giardia vaccine available for pets before this novel one. What were the difference between these vaccines?
Yeah, that's really true. The Giardia vax, which was developed in Canada and then sold by Pfizer Animal Health, is a protein of is a formulation consisting in a homogenate of trophozoites of unknown procedence, which are 17 papers, I think, saying that this vaccine does not work in pets. The company continued selling that vaccine now under the name of Zoetis, which is the name of Pfizer Animal Health that now is called Zoetis. And Zoetis is the company that licensed my vaccine for generating a new vaccine to replace Giardia. Unfortunately, they bought the license and kept the license for 6 years without doing anything. And now, just last month, the CONICET was able to recover the license from Zoetis to be able to pass on to another companies to develop an effective vaccine against Giardiasis in pets.
Okay. The next question from Victor is, the VLP based in VSP proteins was designed to resist the proteolysis in the gastrointestinal tract, as you show. What do you think that gives the protection characteristics to these VSPs?
I still believe that to answer correctly that question, we need to get the real structure of the molecule, something that we are working on that. But so far, I think that this characteristic is given to the protein by the presence of these system XX system motifs together with the metal coordination. This is all what I have to say about it. These are the results that we have, but until we don't get the molecular structure by X-ray crystallography or cryo-EM, we won't be able to make sure what the real characteristics of the protein are making this protein so fascinating.
Yes. A question from Bartira Bergman. Have you tried vaccination with live parasites expressing the whole VSP repertoire followed by 1 week later with oral metronidazole to abrogate infection?
Yeah, we have done a whole bunch of experiments a long time ago. That was my intention during this presentation, is just to give the story of the research that we're doing during decades. And in the Nature Medicine paper published in 2010, you can see all these results. Originally, before purifying the VSP from the parasite surface using the particular monoclonal antibody, we were infecting the animals with trophozoa expressing the entire repertoire of VSPs, and then challenge the same animals later with and without, with and without treatment, metronidazole treatment, for example, and challenge these animals with strain expressing a unique VSP on the surface, or from cysts coming from the clinic in which we don't know the VSP these cysts are expressing once exist. And all these results are published as supplementary material in this Nature Medicine paper.
Okay, could you answer all the questions?
Sure, no problem.
Oh, thank you. Dr. Ana Paula asks, one of the main problems for oral vaccination is the higher dose compared to the traditional immunization. Have you optimized the concentration?
Sure. There are two problems with oral vaccination. One problem is that the intestine, as considered by most of the people, is a tolerogenic organ. So the large intestine is tolerogenic. So it's meant not to produce an immune response to the bacteria and microorganisms that make our flora, intestinal flora. So we have to make clear that we are going to vaccinate on the upper small intestine. We have demonstrated in previous experiment and published experiment that the VSPs of Giardia attach to the mucosa of the upper small intestine and remains there attached for hours. So the concentration that we need, of course, can be higher than vaccine or given orally. But if we can produce a capsule to easily show how much of the antigen we are going to use, it's going to be fine. It's not going to be any problem. And taking in consideration that the VSP attached to the epithelial, to the mucous layer of the upper small intestine and the epithelial cells of the gut, it doesn't matter if we use a low amount. We're using nanograms of proteins for vaccination. We can this protein can remain there for a long period of time. We measure that period up to 12 hours. But of course, we have to make additional experiment for that. But we believe that the easy production of these VLPs, the versatility of the platform, together with the production of the antigen in the right conformation, we are able to get a very strong immune response by vaccinating on the upper small intestine. If we want to vaccinate against allergies, for example, allergy antigens, we would like to use the large intestine, not the upper one. So this theory of how this works can be tested easily in the future, and we're glad everybody that wants to try this system, not only for vaccination, but all, against pathogenic microorganisms or tumors, but also to other allergens that can cause problems, and inducing a tolerogenic response in the lower intestine could be highly beneficial.
Okay. We have a question from Dr. Guadalupe Ortega Pierres. How are the VSP exported to the surface of the parasite? Which are the immune mechanisms that are activated in the host and that induce protection? How are the VSP organized in the surface of this parasite together with other proteins?
Oh, that was something that was published in the late 80s by Franz Gielings and David Reiner, I guess, if I'm not confused, in which they show that the interaction among the molecules on the surface of the parasite was due to the cysteine XX cysteine motifs. It was very clear at that time that there are no free sulfide drug groups on the surface of Giardia. So there is no doubt at this point that that the VSP hold together by some interaction, maybe intramolecular or intermolecular disulfide bonds or metal chelation. However, I don't think that the VSP make a coat like an umbrella that can avoid other protein to be seen by the immune system. In particular, because of our results with the hemagglutinin antigen from the flu, you can see that even the antigen from the flu is protected from degradation, a different pH, different tough conditions. The hemagglutinin can be seen by monoclonal antibodies, for which we label the particles for immunoelectron microscopy or for immunofluorescent microscopy can be seen, and also by the fact that the validation of the VLP that we performed was using the inhibition of hemagglutination assay. So in that case, everything suggests that the protein is available there. The question is how the VSP protect the other protein? We still don't know. We are based on the previous experiment of Gielin and Reiner, and with our experiment with the treatment of the disulfide disrupting agents. And in those cases, we lost these properties. So it's very clear too that that the VSP are not a general inhibitor of proteases, because when we use intestinal extract to test if the intestinal extract destroys the VSP, we have many different kind of proteases present. We have carboxypeptidase, we have aminopeptidases, we have endopeptidases, we have different kind, and we know that the inhibitor of proteases are very specific for the different kind of proteases. So it's something that we still don't know, but we have a very good idea that this kind of protection or this kind of immune response is produced by the presence of multiple cysteine XX cysteine motifs.
Okay, thank you. I have a question. Do you answer me? And do you believe that this set of VSP proteins, of course, in the oral vaccine could be helpful in chronic giardiasis?
Yes, I have no doubt about it. The question is we haven't tested. What I think is, if we have such a beautiful results in journals, we are not the target of Giardia lamblia or Giardia intestinalis. Are not the real host. And we have a good immune response, and we have a good protective response in journals. And in humans, I suppose it's going to be better, because Giardia is a pathogenic of humans. So I really think that it's potentially important to, in the future, when our oral vaccine or other that we are developing at this time by other means, we can vaccinate patients that have chronic giardiasis in order to generate a humoral immune response on the intestine that can recognize any VSP that these parasites are exposing. We think can be used also therapeutically.
Okay, thank you. Fabio, please come in.
Hello, Professor Luján. Thank you very much for this beautiful presentation. I would just have 2 questions, if I could ask, and then we maybe could...
no, I have no problem at all.
All right, great. I'm not sure. Okay, let's see. So 2 questions. So first, about the molecular mechanism that Giardia is using through the RNAi pathway to regulate VSP expression, do you have any more hints on how it's actually operating at the molecular level? And also, so how diverse are the VSP haplotypes of the strains around the world? Is that the ones you're using, is that a good representation of the diversity we'll find in different geographic areas?
I'm not sure. Then we can go back to the second of your question. Let me focus on the first one. When you try to send a paper to Nature or Science for publication, you are not in the power of determining the title of your publication. So in that paper, it was clearly antigenic variation in Giardia regulated by RNAi, something that is true. The point is, it's not completely true. Our title was totally different, not such some strong one, because we already have some preliminary result in a paper that we published a little bit later, showing that there is a combination of factors, some epigenetic factor, some RNAi factors, which playing together the regulation of the expression of a unique VSP from a repertoire of about 150. And so it's not so simple to say it's just RNAi. There are many other factors, but we know that RNAi and small interfering RNAs that are produced in Giardia all the time, as are produced in other microorganisms like T. brucei, like Toxoplasma, like Plasmodium. All of them, if you sequence the small RNAs of this microorganism, you will find a lot of small interfering RNA that target not only surface antigen, but many other genes at the same time. This is one point. So when you have a small RNA, you also have you play a role with a lot of chromatin modifying enzymes that acetylate histones, the acetylate histone, methylate some lysines in some position of the histones. So there is a very complicated mess, and which is clear is that most of the antigen are ready to be expressed if the conditions are right. We are working on that very hard. We have a whole bunch of transcriptomes of many different Giardia from many different organisms, like natural infection in dogs, in cats, and humans, and we are assembling the entire repertoire of that. I don't know if I understood correctly your second question, but I think it's related to that, right? The fact that how we know if we use the WB isolate and we can have an immune response to another isolate, for example. Is that right?
Yeah, I was basically going to this direction. I was wondering if there are some more genomic studies from strains around the world where you can actually look at how diverse are the VSP haplotypes on those strains.
Yeah. There are many. In fact, many Giardia genomes have been sequenced not with the quality of the WB-C6 clone of Giardia, which is the one that we use then to analyze all these variants. But remember, there were some papers in the past that Giardia have more or less 350 VSP genes in the genome. Then there were some publications by CC1 that said that Giardia have only 60, 70, that just fix it with the number of microRNA he want to fix too. But if you analyze the VSP from many, they are really similar, with the diversity among them that is present in only one isolate. But the question is, when we vaccinate with the WB isolate, we have 100% protection if we challenge with the WB isolate. But we have only 70% protection we challenge with the GS isolate, which it belongs to assemblage B. So to get the perfect vaccine covering all the potential VSPs that can be present in parasite infecting humans, we have to work on making a combination of assemblage, just by disrupting antigenic variation by any means, just RNAi, disrupting RNA pathway, disrupting epigenetic mechanism controlling the expression of the VSP, etc. But can be done.
Thank you. Okay, I'm not sure if anyone else wants to address a question. Otherwise, Professor Hugo Luján, on behalf of the graduate school from our Biophysics Institute, I would very much like to thank you for this beautiful presentation. And I believe that Professor Vanderlei Souza would like to make some final comments. Vanderlei? Vanderlei, are you there? Okay, he's turning on his microphone. Vanderlei, we cannot hear you. I believe that we have some problems with Vanderlei's microphone. Okay, so it seems that, okay, he's trying again, maybe. I'm just following like the turning on and off of the microphone from the side screen. Okay, Professor, I think I will try to say thank you on his behalf as well. I'll take this freedom right now. So thank you very much. Hopefully we'll be able to keep in contact. I'm sure that Vanderlei will reach you later. But thank you very much for this beautiful presentation. Thank you very much for your time and for taking part of this event. And we are very honored to have you here, even if it is under remote circumstances.
Thank you and very good luck.
Okay, Professor. I think I will try to say thank you on his behalf as well. I'll take this freedom right now. So thank you very much. Hopefully we'll be able to keep in contact. I'm sure that Vanderlei will reach you later. But thank you very much for this beautiful presentation. Thank you very much for your time and for taking part of this event. And we are very honored to have you here, even if it is under remote circumstances.
Thank you and very good luck.
Okay, Professor. I think I will try to say thank you on his behalf as well. I'll take this freedom right now. So thank you very much. hopefully we'll be able to keep in contact. I'm sure that Vanderlei will, under remote circumstance.
Thank you. And